This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Since the very beginning of plant breeding and domestication, plant breeders have been relying on the natural occurrence of new traits via spontaneous mutation in the population to produce new plant varieties. When a new interesting trait occurs, a breeder can spread it in a population via mass selection or incorporate it in a new purebred line with controlled crosses. Unfortunately for the breeders, the occurrence of natural mutation is very low, and mutations that are actually of interest for plant breeders are even lower. But since the discovery of induced mutagenesis in the 1920s, it is possible to induce mutation in plants at much higher frequency than their natural occurrence. Depending on the mutagen agent, induced mutations can be 1,000 to 1,000,000 times more frequent than naturally occurring mutations. Mutations can be induced using mutagens, such as DNA-altering chemical compounds, all sorts of radiation, X-rays, atomic radiation, or UV light. More recent techniques of mutation induction include the use of ion beams, which can cause deletion of large DNA segments, or even space breeding, where plant material is literally shipped into space and cosmic radiation, zero gravity, absence of atmosphere alter their DNA. All these mutagens damage the cell's DNA to different extent, and while the cell is trying to repair the damage, its own repairing mechanisms will cause the actual mutations. There are different types of mutations that can be induced. Substitutions are mutations in which a single DNA nucleotide is replaced with another. Deletions are the elimination of one or several nucleotides. Insertions are the introduction of a single or a group of nucleotides in the DNA sequence. Inversions happen when a portion of the chromosome is flipped, turned around, and then attached again. An important thing to remember is that each chromosome is uniquely affected by mutagenesis, so a new mutation, whether induced or naturally occurring, is originally in a heterozygote state. It is present in one, but not in the other chromosome. Unfortunately, mutation breeding is not as simple as producing an X-mutant or Spider-Man. Although causing mutation with a mutagen is fairly simple, this is only the beginning of the process. The individuals carrying the mutation of interest then need to be identified via screening, and this is less or more complicated depending on whether the mutation is dominant, recessive, how it affects the phenotype, and whether it can be identified by phenotyping or also by genotyping. If the mutation is dominant and is visibly expressed in the phenotype, it can be simply identified by inspecting the phenotypes of the individuals exposed to the mutagen. The effects of other mutations are not directly visible, but can be detected by applying a screening agent. For instance, if a dominant mutation confers pesticide resistance, simple application of the pesticide will eliminate all plants that do not have this resistant mutation, since only plants that carry the pesticide resistance can survive. If the gene coding for the trait of interest is known, then the mutation of interest can also be identified by genotyping the plants and identifying the individuals that carry the mutated genetic sequences. Once the mutated plants have been identified, the mutation needs to be made hereditary. If a mutation is induced in a seedling or a fully grown plant, each of its cells can have a different type of an induced mutation. However, these mutated cells are somatic, and the mutation is not hereditary, meaning it cannot be transmitted to the offspring. The only way for a mutation to be hereditary is if it occurs in a germline cell of the pollen or an ovule. In this case, all cells of the offspring will carry the mutated gene, including its own germline cells, so the mutation will be hereditary. But even somatic cells can be grown into an adult plant using a cloning technique called plant tissue culture. the cell or tissue presenting the desired mutation is excised from the plant and placed in a nutritious environment. This environment is supplemented with a growth hormone that will stimulate cell division and differentiation into a fully grown adult plant that expresses the mutation and can transmit it to the offspring. Alternatively, mutations can be induced directly in a germline, such as pollen or ovules. The mutated pollen can then be used in a controlled cross, and the offspring resulting from the cross will carry the mutated gene. There is another way to produce plants carrying the pollen mutation without recurring to pollination. Using a chemical treatment, the genome of the pollen can be doubled to obtain what we call a double haploid cell. It is a diploid cell that contains two identical copies of a haploid set of chromosomes. Double haploid cells can then be stimulated to divide and differentiate in a fully grown plant that is homozygous for the mutated gene, and this gene will be hereditary. Detecting a mutation is more complicated if the mutation is recessive, which is often the case. If the mutated gene is recessive, its effects cannot be phenotypically detected by simple observation of the heterozygote plants, nor it can be screened by applying a pesticide or a herbicide. This is because the effects of the recessive, mutated allele are masked by the expression of the dominant, non-mutated allele. So how do we detect a recessive mutation? If the gene of interest is known, the plants can be genotyped after the induction of mutation in order to identify the mutations of interest. But what happens if we don't know the sequence of the gene of interest? If the mutation is recessive and hereditary, it can be revealed in a homozygote recessive offspring. After inducing a mutation, the plants are left to self-pollinate. If a plant has the desired recessive mutation, about a quarter of its offspring will inherit two recessive alleles and will express the mutation in their phenotypes. If the plant does not have the recessive mutation of interest, none of its offspring will express it. If the recessive mutation is induced in germ cells, its identification can be accelerated via the production of double haploids. Simply double the genetic material in all irradiated gametes, and after growing plants from these double haploids, those containing the doubled recessive allele will be easy to identify phenotypically or via screening. So once all the problems with identifying mutated individuals and rendering the mutation hereditary are solved, the last thing to do is to eliminate all undesired mutations from the genome that occurred at the same time. This is done in the same way as for the selection of purebred lines, through a series of controlled crosses. To sum it up, mutation breeding is a technique that accelerates the occurrence of mutation in the DNA by inducing high mutation rates in plants exposed to a mutagen. The mutations are induced at random, so the desired mutation needs to be first identified by genotypic or phenotypic screening, and then incorporated in the germ cells of the plant using techniques such as controlled crosses, double haploid production, or plant tissue culture. Once the mutation is successfully identified in an individual, a series of controlled crosses and selection will eliminate the undesired mutations that occurred at the same time. Let's see a couple of examples of plant crops produced with mutagenesis. A barley variety called Diamant was created by mutagenesis in Czechoslovakia. This mutant variety has high tillering ability, shorter stalks, both of which render the plant more robust, and also higher yield than any of the varieties grown in the Czechoslovak fields at the time. By 1972, almost half of the barley fields in Czechoslovakia were planted with the Diamant variety, and even today, some of the Diamant characteristics are introduced in new barley lines using controlled crosses. An atypical example of a plant obtained by mutation breeding are several lines of bluegrass obtained from space breeding. They're characterized by reduced growth compared to other bluegrass lines. This reduced growth is very advantageous for their maintenance. Bluegrass is typically grown in mounds and requires frequent mowing about once a week. Because of its reduced growth, the space mutant bluegrass can be mowed less frequently, thus saving time and effort dedicated on its maintenance.